Boy, I love retirement.
I just feel old and my joints hurt.
Mine don't.
But we're the same age. How can this be?
Did you know how old you are on the outside might not be how old you are on the inside? But how do you measure how old you really are? To answer this question, we turn to the clock.
The process is simple. First, take a couple drops of blood or a urine sample. And send it out for analysis.
Age measuring tools are now commercially available through companies such as MyDNAge and True Diagnostic. These tests are built around epigenetic clocks. But what is an epigenetic clock, and can it really measure aging?
So how do you determine biological age? Many approaches rely on measuring gene regulation. To understand this process, let's first talk about how proteins are made and the central dogma of biology. The central dogma describes how DNA is transcribed into RNA, which is translated into proteins that perform nearly all cellular functions. Molecular processes regulate these important phenomena at every level of the central dogma, determining which proteins are present in the cell. One method of regulation is called epigenetics. Epigenetics describes the physical and spatial, not sequential, modifications made to DNA that impact the transcription state of genes, and therefore if they are turned into their respective proteins. One specific epigenetic method of regulation is known as methylation. Whether a gene is transcribed relies on an upstream sequence known as a promoter and a segment within the promoter known as a CPG island. CPG islands are stretches of DNA characterized by a high density of cytosine and guanine nucleotides. The specific type of modification measured in many epigenetic clocks is known as methylation. When CPG islands are unmethylated, transcription happens unencumbered. However, when these regions are methylated, transcription no longer proceeds, halting protein expression. As we age, more genes become silenced through methylation. Researchers have developed methods to accurately predict biological age through measuring methylation status at many sites in the genome.
The pioneer of this field, and first to make the association between epigenetic status and age, was Steve Horvath, who combined the use of sequencing technologies with computational modeling to select 353 CPG sites that best correlated with age in his training data. Since then, many different clocks have been developed and include variable numbers of CPG sites. Some are correlated with specific age cohorts or tissues, while others work across age groups and sample types. But these clocks share one common theme. They are the most accurate predictors of aging in healthy individuals. Most have an error rate lower than 5 years, proving better than other commonly used metrics, such as telomere length. You mentioned multiple sample types could be used. Does that mean this would work for our pet frogs? Jeremy?
And Gretchen?
Yes. Scientists can now measure the epigenetic age of a variety of animals, including frogs, such as your Gretchen and Jeremy, as well as cats, dogs, and naked mole rats. The conservation of epigenetic changes with age across organisms and even evolutionary branches indicates that epigenetics may be at the core of aging. Some view this as evidence in support of the evolutionary senescence theory of aging, which states that selective pressures for positive traits are weak after reproductive age, leading to changes in epigenetic associated gene control. Others view epigenetic changes as the driver of aging itself and not merely a byproduct of evolutionary pressures. These scientists have posited the epigenetic clock theory of aging.
Methods of measuring biological age have continuously evolved since the establishment of the first epigenetic clock and are now classified by generation. First-generation clocks, like Horvath's and Hannum's, focus on predicting chronological age using specific DNA methylation sites. Second-generation clocks, such as PhenoAge and GrimAge, go beyond age prediction to assess health risks and mortality by incorporating clinical biomarkers instead of or in addition to CpG methylation and lifestyle factors. Finally, third-generation clocks rely on the most diverse set of biomarkers yet to predict the rate of aging.
Now, let's check back in on our age-measuring friends.
Dear, I'm going to get the mail. We heard back from the clock place.
Oh, good.
Wow, I'm only 60. I'm gonna
What?
go on an adventure.
What? Those scientists say I'm 94.
That seems a little too old. We're only 75.
How can this be?
Well, higher predicted age has been associated with activities that are known to accelerate aging, such as smoking or aging-associated diseases such as type 2 diabetes. Thus, your results may indicate you are aging at an accelerated rate.
Doc, what can I do to slow down this aging? I gotta go on that adventure with my wife.
Well, there is no foolproof method to slow aging. However, some interventions have shown promise, such as exercise and caloric restriction. Also, several drugs and small molecules are under investigation. However, before you change your lifestyle, you should keep in mind that the link between accelerated aging and a high predicted age is based on correlations to aging risk factors, and a causal connection has not been established.
Thanks, Doc.